So next on the agenda is a report on the H3Africa program. And H3Africa has been a common fund project for 10 years. It has reached the end of its 10-year cycle. And NHGRI had a prominent role in many aspects of this project, including the peer review of multiple rounds of applications and the renewals, a ton of time on the part of grants management officers and program directors to help manage and coordinate this program. So we thought it was fitting to have Jennifer Troyer, a program director in Genome Sciences, give kind of a summary of the project. So Jen, take it away.
Thank you, Rudy, for that introduction, and thank you. It's wonderful to see all of you in the room, and thank you for those of us joining online. It's my really great honor to be able to present about this program. As Rudy said, it's a common fund program, and it has been led by NHGRI for quite a while now, and I am going to ask you to put on your time travel hats and think back for over 10 years, because one of the things I'm going to talk about is some of the features of a common fund program. We've talked a lot about how NHGRI carries more than its weight in common fund, and there are some particular features about common fund programs that are unique across the NIH. And so one of those, they come from the Office of the Director, and as such, they're supposed to be useful for all of NIH. So all the NIH institutes and centers are supposed to benefit from common fund programs. They are catalytic and transformative, and so they're supposed to take an area of science and really move it forward in significant ways. And they are time limited. So sometimes 5, but usually as much as 10 years, but no more than 10 years. And we'll get in the time frame in just a little bit. And in that time, they're supposed to do something that is high risk and high reward. All right, so listed here, I'm not going to read all of them, but this program started with NHGRI before I started, and many people have come and gone and been very active contributors during that time. So I appreciate all of their work. And then, as I said, Trans-NIH, there are our working group members across many institutes and centers. This is the current group of folks. There are also past ones that have contributed over the years. And then H3Africa is not only an NIH program, but we have funding partners internationally. And so there are program staff at the Wellcome Trust and at the Science for Africa Foundation who also work on the program. And then over the years, we have been guided by a panel of international independent experts, including Charmaine, who's still here somewhere, I think, who've really helped the program evolve over the years. So the goal of H3Africa was, and I'm asking you to put on your hats and go back to before 2010, 2007, 8, 9, when it was first coming to the fore as an idea at the African Society of Human Genetics, was to do something that hadn't been done, which was to really enable the genomic revolution in Africa and make sure that African scientists were able to take ownership of African research. And that was, at the time, considered a high-risk endeavor. And so there was to put forward scientific goals, of course, to do genomics research in Africa, but then also some capacity development goals, both for human and research infrastructure capacity, but also building collaborative networks and allowing African scientists to collaborate with each other and not always rely on a funder from a first world country. So One of these pictures you've seen before, but I think it's worth seeing again. So back in 2010, this program was launched at official press release with Bangani Mayosi and Mark Walpole from Wellcome Trust, and then some familiar characters here, Eric Green, Francis Collins, and Charles Rotimi. And the first calls for applications, or RFAs, went out in 2010, and the first awards were made in 2011. And then the consortium came together to publish the first marker paper, where the consortium, the group of people working on this, laid out for themselves some goals. And among those were increasing the number of publications in high-impact journals that had first and last authors from Africa that were about genomic science. Also, the ability to come in for competitive funding opportunities and obtain competitive funding. And there are several others. I'm not going to read them all, but I really did want to highlight those. I would also point out that as all Common Fund programs have, there was a hard renewal process after 5 years that involved quite a bit of oversight and review to see that the program was being successful and should continue, and it did. And so new rewards, what we call phase 2, were made over the next several years. And I will also point out that in the last awards were made right before some big, huge event happened, the global pandemic, which one could argue is still impacting everything that goes on and everything we do. And so while officially the program came to an end, and Rudy had pointed that out, that this is time limited and we're done, there are several awards that are still in no-cost extensions, and a lot of work that's still going on, and a lot of productivity that's still coming out of this program, and will be doing so for years to come. So here we are at that end of those no-cost extensions, and I just want to say our last in-person meeting was held in Cape Town a few months ago, and that's a bunch of RPIs waving goodbye to the program. So I'm going to give you the history of the program, and then we'll talk a little about what's moving forward and what's next. There were several types of projects, and these were research projects that were given to African institutions. In the case of the collaborative centers, they were required to collaborate, but not with US or European investigators, but with other African investigators. So that's what the collaborative centers were really to develop networks of collaborative projects. Of course, all projects were also welcome to collaborate globally if they so chose. In addition, there were several types of infrastructure projects, biorepositories, informatics networks, a coordinating center, and then in the second phase, Fogarty International Center joined NHGRI as lead institute and put in some training programs that I'll also talk about. The research, however, was what we like to think of as investigator-initiated. So African researchers were welcome and invited to come in with whatever research projects they were interested in that had to do with genomics and human health. And so these came in, were reviewed, awards were made, and there were several types of diseases and conditions and human conditions that were in the network. And so projects kind of clustered together naturally into some working groups that focused around disease areas. So the mental health working group, there was a microbiome, CVD, rare disease, and infectious disease working groups formed naturally from the projects. But as time progressed, additional connections and links between these areas were made. And so we really ended up with quite a network of researchers who are collaborating on a bunch of different things. And I'll just give a few examples. Sickle cell disease project and the malaria project had some natural affinity. Same with the kidney disease and trypanosomiasis, kidney disease and sickle cells as well. So additional connections and collaborations and networks were formed. All right, so what does the consortium look like in terms of the map of Africa? don't worry about the colors. Each of those sites represents somewhere where there are H3Africa researchers working. Some of these are hospitals, collection sites, clinical sites. Some of them are analysis sites, informatics nodes. I'll talk a little bit more about those. And some of them, of course, are laboratories, as well as places where outreach was done to the community with ethical, legal, social implications research. And so an important part of H3Africa was really to make sure that we were doing outreach to communities, to governments, to local regions, to promote the use of genomics in healthcare and in health research. And so each of our consortium meetings that happened twice a year in person before the pandemic hit, and then for several years virtually, and then we've just had our last ones in person again, happened in a different place in Africa, and we tried to rotate sort of around the continent as much as possible. So the top meeting is the very inaugural meeting, which was in Addis Ababa, Ethiopia. The middle one was one of our last meetings before everything shut down, and that was in Kigali, Rwanda. And then the last picture there is our first post-pandemic, or at least our first post being able to be in person again post-pandemic meeting, which was in Abuja, Nigeria. And so, as I said, there's all the people in the consortium came together to form different types of working groups. There were several working groups where every project was represented to make the policies, community engagement, the data and biospecimen sharing, publication policies, et cetera. But then there were also these scientifically motivated working groups. One of those was the genome analysis working group, another was the phenotype harmonization working group to work all across the consortium, and then some that were focused around specific disease areas like HIV/AIDS. Again, we had a steering committee that was made up of the PIs and an independent expert committee of folks that were advising. Outcomes from this, as I said, working groups came in two flavors, sort of policy and guidelines, and then research tools and products. And so there are guidelines for community engagement, for getting informed consent, for feeding back findings, and those are all published and available and actually used in some cases by the governments in the countries, and in other cases just by other researchers, but they've been widely distributed and used. And then there are the research tools and products. So there's harmonized phenotypes. There are 26 phenotypes across all the projects that have been harmonized, and then some specific ones like in cardiovascular disease that have been harmonized across several of the projects. Training programs that have been shared across the consortium. I'll talk more about that a little bit, but also the personnel that have been trained throughout this entire project. It's a huge group of people. And then some cross H3Africa studies on population genetics, on pharmacogenomic variants, on HLA types, blood traits, et cetera. And the genotyping array, which I'll, again, focus on in a few slides. So that's for sort of the working groups of the consortium, but there's also, of course, individual projects, research projects that have been going on that have produced a large amount of data, demographic information, anthropometric data for several of them, and then depending on the project, disease and health-related phenotypes of various sorts. The genomic data also comes in a variety of flavors, so not every project within the consortium has collected the same type of data. There's whole genome, exome, some targeted sequencing. Many, in fact, over 50,000 samples now have been genotyped on the H3A chip. There's also some epigenetic data, some transcriptomics. A couple of the projects are focused on microbiomes, some of them in combination with the human genome, some of them microbiome on their own, and those have both mostly patient and sample phenotypes associated, but also some environmental data and some environmental microbiome studies as well. And those come either 16S or metagenomic, depending on the project. And then the ethical, legal, social implications research has quite a bit of qualitative data available from focus groups, surveys, workshops, et cetera. Of course, a huge outcome is publications, as with any NIH-funded consortia. That's one of our measures of success. We're actually just tipping over the 700 publications mark. And these, again, come in a lot of different types because there are so many different things going on within the consortium. Some of them come from projects, others from working groups. But data and methods, of course, disease risk is a huge portion of them. That's that green section, but also quite a bit on capacity building, on health equity and social determinants of health, participant and community engagement, training, et cetera. And I am going to show you a few papers. I do not expect you to read all the details. These are just example papers that have been published recently, and I want to point out a couple things. They're in respected international journals, as was one of the goals. They have a variety of different topics. So this particular one is cardiovascular risk factors, but they have African authors. And I will say, for the most part, many of them have other collaborators that are global, but for the most part, the vast majority of the authors are African, and they're not in one place in Africa, they're spread across Africa. And that's true for the pharmacogenomics, for the perspectives on genomic data, for developing new analysis methods. All of the papers that are coming out are African-driven, which is what we were hoping for. This is our sort of flagship publication that came out a couple of years ago that we were proud to share this issue of Nature with the NHGRI strategic plan paper. So get that issue, it's a great one. And it examined high-depth African genomes, sort of across the consortium, across the continent. There was a concerted effort to fill some of the gaps in terms of populations and people with genetic similarities that had not been sampled before. And led to this also commentary, this nice editorial about how African people should be driving their own genomics agenda. They also got a cover on Nature that has this lovely beaded structure plot of all the genomes from the H3Africa consortium that one of our scientists, who's also quite talented, made these beads. As well as publications, as well as careers, there are several resources that are now available for use by anybody. And so H3A BioNet is the bioinformatics network, this infrastructure that has been established there are 35 sites in 17 different countries in Africa, where there is local expertise and there are competencies that they've all established jointly and are able to accomplish in genome data analysis and workflow development, data management, et cetera. Along with, of course, the people that are doing these things, there is the infrastructure to do them. In some places, that was very challenging to develop, but it's there now. And then throughout the 10 years, this is only a small smattering of the accomplishments of H3A BioNet, but I do want to point out that they were really instrumental in designing the original array and making sure that African variation was tagged correctly and that there were population-specific reference panels that could then be used for imputation, and they do run an imputation service that is geared towards the appropriate population reference. And they have available SOPs and Docker containers for all the H3Africa analysis pipelines that people can use. Along with this, there's a considerable amount of training available, and all of this, again, is packaged up and online and available along with training data sets for people to use if people want to establish their own infrastructure, pipelines, competencies. All of that is available online. And then H3A BioNet is also our data storage and transport and access portal. And so it houses the H3Africa data and biospecimen catalog that will tell you all the data that is available, where you can find it, what the data use ontologies are, and which of those genomic data have corresponding DNA samples in the H3Africa biorepositories. Speaking of repositories, another resource that is there. And so while the samples that they're storing for H3Africa are DNA samples from the studies to do the genomics, they have really developed many different competencies, the ability to deal with many different sample types used in biomedical research. They meet international standards and participate in international conferences and biorepository groups, and really can support any research project, up and coming or past research projects, in sample collection, processing, storage, shipping. The other thing that's been great is, so I should say all our sample sites, collection sites, and this was not by design, but by the way in which projects came in and were funded, are sub-Saharan. So we have repositories that cover 3 major sub-Saharan regions of the world. And they've worked out, which took quite a lot of doing, affordable, reliable courier shipping routes that and have sample MTAs, import-export permits. They know how to get these samples across the borders effectively and efficiently. So again, useful for future projects. I've mentioned training a lot, and a huge part of H3Africa has been considered to be training. So the official training programs that are Fogarty run have established or enhanced master's, PhD bioinformatics programs at many universities across Africa, and these have supported over 100 graduate students at this point have graduated or are graduating through these specific training programs. But in addition, all of our projects are very training focused, and hundreds of graduate students, postdocs, young investigators have also been trained within the research projects and gotten their degrees. So one of the things that I didn't mention on the BioNet page, but that BioNet has been instrumental in and has been a great product from that, has been this introduction to bioinformatics training distance learning classes. So they're offered massively parallel classrooms across Africa, and thousands of students take those every year. And then there are other courses, workshops, webinars, all sorts of things that have been run either by the working groups, by the coordinating center, by BioNet, and those are all available on the website or on our YouTube channel, or both. Another thing that we've done with training has been, for most of the meetings, starting, I think, in about the 4th or 5th meeting, the in-person meetings have given fellowships that have been competitive. So trainees have applied to come to the meetings and have been able to present either speed talks or posters and get career development opportunities like grant writing, manuscript writing, panels on various career paths. At those consortium meetings. So we've really tried to focus them on trainees. And at the very last one, the consortium chose 4 emerging leaders. These are all up-and-coming scientists who got their PhD and then did their postdoc within the program, and now are early-stage faculty at various places. So look out for these people. They really are up and coming, and you'll be seeing them in the future. And then I, mentioned there was this pandemic, and the H3Africa investigators were ideally placed to respond. And so I have to give a shout out to Christian Happy, whose project is in sequencing viruses, unknown viruses, fevers of unknown origin, and so had the BCL laboratories and the sequencing capacity in place on the ground when COVID hit Africa, and so was responsible for sequencing the very first African SARS-CoV-2 and continually throughout the pandemic sequenced a lot of the samples that came from patients across Africa through the African Union, the CDC. And then Tulio de Oliveira, you may have heard of, leads the Network for Genomic Surveillance in South Africa and was responsible for the early detection, characterization, and sharing information about the new variant of Omicron. And then I have to say, I have to give a shout out to our folks really across Africa, particularly the biorepository PIs and staff. So you can see they quickly pivoted to being centers for collecting samples and testing and providing QC and testing all the test kits that were coming in to their various regions. And so they were 24/7 operating throughout the pandemic at the biorepositories. And then also our community engagement networks. So every single H3Africa research project had embedded community engagement in it. And so they were on the ground, able to be out in the communities talking about wearing masks, about distancing, and then later about testing and about vaccines. In addition, several of our PIs got pulled by their governments to advise them about what to do in the face of the pandemic. As well as helping their national governments, our PIs are now embedded in many of the international collaborations. I think you saw about the collaboration with ClinGen in Eric's director's report. They've been talking to the Pan-Genome Reference Consortium about including African genomes in that. More of them. There are, of course, several already. And then also working with the African Society and the American Society of Human Genetics to make sure we're traveling and representing diverse people in those meetings. And a couple of our H3Africa investigators are now LMIC, Low and Middle Income Country champions for the Undiagnosed Disease Network International. In addition, I think the NIH does not see funding research in Africa as such a risky proposition anymore. And so this is another common fund project that started recently, and it is about harnessing data science for health discovery and innovation in Africa. It's called DSI Africa. And the idea really is to, again, advance the ability and the infrastructure to use data science for innovative health discovery, and it includes government and industry partners. And so it differs from H3Africa in that, but some of the ways in which it's modeled on H3Africa is the awards are made directly to the institutions in country, that it is collaborative, that there's embedded ethical, legal, social implications research. And then, of course, because as we all agree, right, genomic data is one of the important data types, there are a lot of our investigators also involved in these new data science projects. In addition, other funding is available, of course, and one of the goals was to have the scientists in Africa be competitive for normal NIH funding opportunities, and you can see that they are coming in for RFAs, for other consortia, becoming involved in those, and also just getting R01s and R21s and R03s, things like that, and from institutes and centers across the NIH, which is really fantastic. And then, of course, NIH is not the only biomedical funders out there, and they're getting funding from a lot of other sources as well. So looking forward, I think we're in a really different place. And in fact, I would say looking forward, it's really hard to look back on to that time 11, 12 years ago when this was seen as something that might not work or that wouldn't be a good idea. It was considered high risk. And those of you who were there at the beginning might remember it was a little bit of a hard sell for NIH even. And now you can't sort of think about doing genomics without including African genomes and without including African scientists, more importantly, I think. And the World Health Organization is now thinking about genomics for global health in low and middle income settings. As Charles talked about, there are plans for these networks of genomic centers of excellence across Africa and some audacious, bold ideas about sequencing 3 million African genomes. So a lot's going on, a lot's to come. I just want to say that none of it would happen without the wonderful researchers. Here they are at another consortium meeting. And I will say every single one of the people that I've worked with in this consortium here and in Africa has the spirit of our beloved Pangani Mayosi, who's always said, lift as you rise.
Okay, thank you, Jen. Questions for Jen about H3 Africa?
While council members are thinking of things they may want to ask, I should point out that, when NHGRI takes these things on, these common fund projects, I say, they are a lot of work, but we also really try to make sure we are doing this NHGRI style with the best possible leadership because it is pivotal. And I would say I really believe that one of the key successes of H3 Africa is early on we had Jane Peterson and Mark Geyer, and they handed the baton to Jen, and all of them, and particularly Jen, most recently just done a spectacular job, very heavy lifting, really complicated projects. And I guarantee you it would not have been as successful if it wasn't for NHGRI bringing excellent program staff and leadership to it. So Jen deserves a lot of credit, as do Mark and Jane. And the Wellcome Trust has been wonderful to work with, and others at NIH who have been helpful. But now Jen, in particular, as Francis Collins has been spending some of his time trying to develop the sort of next set of initiatives that might come out of that, probably outside of NIH or partially inside of NIH, doing this. He has Jen on speed dial, so I know he just goes right. And Jen's been very helpful to Francis in some of these pursuits that she mentioned and Charles Rutimi mentioned.
Yeah, and I'll say a shout out to our grants administrative branch because these were not easy awards to make.
Yeah, I will definitely say that. Think about an entire consortium where almost none of the applicants, some of them had, but most of them had never had an NIH grant before.
Yeah, another way to say it is from the very beginning, we knew this was going to be high risk, not just for the science, but even for the science management, a lot of the sociological. This was like swimming upstream constantly at every front.
Okay, I've got Gail and then Lynn.
Yeah, more of a comment in that vein. I was actually on the original committee in 2011 that reviewed the first set of proposals, and the pitfalls and alternative strategies sections were just so illuminating. It's like, if we don't have power for less than a month, if we don't have power for more than a month, if the government is overthrown. Oh,
yeah.
It was really things you thought you didn't have to worry about in the U.S. And so I really, this was such a big lift and such an important project. So it's incredible to me 10 years later to look at the success of this project. Congratulations.
Yeah, the tenacity of those investigators is really impressive.
Yeah.
Lynn.
Yeah, it's one of those, sorry, go ahead.
So in looking at the map that you showed early on, there's still some very large regions that appear to be unsampled in the continent. And given that Africa has more genetic diversity than any other region of the world.
Absolutely.
Are there plans for further sampling to sort of plug some of those gaps, or is it not what it appears?
No, the gaps are there,
Okay.
and the gaps are there for a variety of reasons, some of them political, right? Some places are just really hard for people even to submit an application or think about genomics when you're trying to avoid a war, right? So, but there are, so in a small way, the original funding for the genomes to design the chip was done in that way, to say, where do we not have any sampling, and are there any scientists who have samples where we can get the right consents? So consents are, of course, another huge issue. Or that we have them, that we can get some representation for those groups. And then So that was a supplement early on for chip design. There was another supplement that was given a couple years ago to now say, okay, now that we've got this project and we can see where we need more sampling, can we get some more sampling there? And so there is in this project called the Agenda going on exactly that, trying to fill some more of those gaps. But then these bigger visionary projects that are talking about genomic centers of excellence throughout Africa will be to sort of start with hubs and put spokes into all those areas where there's currently nothing to try and make inroads there.
I see the virtual hands are up, but I'm going to go to Iftikhar first.
So I had a couple of comments. One was related to the previous presentation. I think if you fund these additional projects, it wouldn't be a bad idea to mandate some population descriptors because of the enormous diversity in Africa. I think that'd be really useful. I know there's an ethno-linguistic ontology that exists, but that would be very useful. The second was, I think the value of genotype data is multiplied if you have phenotype data. And the unfortunate thing, there are no EHRs in Africa. And that may be an insurmountable problem. I mean, I don't think NHGRI is going to solve it, but I wonder whether that's come up in discussions.
Yeah, it absolutely has come up in discussion. And so I'm going to start with the population descriptors, and then we'll talk about phenotypes. So there is a fairly well-developed ethno-linguistic ontology, and that was part of one of the things that the phenotype harmonization working group tried to do early on was to really say, how are we going to determine someone's heritage, right? And how are we going to count that within this? And so looking at those ethno-linguistic groups, and several of the projects actually asked not only, what language do you speak, where are you from, but where were your parents from, what language did they speak, where were their parents from, so going back a couple of generations, actually. I don't know that anybody's figured out what to do with that data, but for some cases it's there. And we did engage with the H3Africa consortium around the population descriptors discussion. We had a whole session at one of the consortium meetings about that, in fact, last year. And they were invited, of course, to give their opinions into the RFI that was out about the population descriptors as well. Phenotypes are harder, and there are some places where there are good electronic health records, and there are actually some places that have a more connected health system than we do. So I wouldn't say it's completely insurmountable, but of course there are a lot of places where there's not, and it's manual entry of phenotypes. And so using REDCap throughout the consortium projects has helped with that, but it's still manual entry.
Nancy, did you have a comment?
Yeah, I have to say, so on some grants that I've reviewed that involved data collection in Africa, it was really impressive how much data could be collected per unit cost. I mean, the costs of data collection, I mean, they could deploy small armies of highly educated and well-trained people to collect data at scale. And at the time, I remember thinking, why aren't we doing more of this? Because the value of having hugely diverse genetic populations in a variety of environments, different kinds of environments, is like the bonus for learning more about the relationships of genetics and exposures for health outcomes. So so I'm really glad to hear that the barriers are lower now, and that that NIH will be considering more kinds of grants from Africa. It does seem it is an opportunity to do well by doing good also in other ways. I mean, it will help us understand things better. So is that the focus of this new round? I mean, I know it looked like it was focused on data science, but for health outcomes, maybe.
Yeah, no, it is not to collect new data, but to take data that exists. So that is a difference for sure. I should mention that NIEHS has been a strong supporter all along and has provided supplements, and they have a HERE program where you can submit samples to get environmental exposure data and things like that on the samples. So several of our H3Africa projects have taken advantage of that and have worked with NIEHS as well to get some of those environmental measures.
But maybe it's time for other NIH institutes to really be deeper in collecting some of that data too.
I mean, Nancy, I think one answer to your question, the way I understand it, is we're probably we probably don't have a crisp answer for what's next for NHGRI, because I sort of heard, I think there's, one thing is we will continue to be as helpful. I mean, first of all, we have to watch what develops. There's various interactions we have in the World Health Organization. By the way, the movement of Jeremy Farrar to the World Health Organization is a very important one. We have and so conversations with him, I think, will evolve, and we talked to him recently, and actually met with him in person recently. So that's something we're keeping our eye on. There are these developments that France is involved in unofficial, I guess officially, but not as an NIH leader, just as an ambassador. We'll see how that works out. I mean, Jen gets pulled into lots of conversations that will continue. And you're right, other institutes are picking this up a little. But I guess my answer, but it would be, I can't predict where NHGRI will be 2-3 years from now. I think we're in a watch mode. We're not ready to jump in anything. I'm not sure what we would jump into, but I share your view that there's great value for learning genomics by, I mean, one, we want to build capacity. That's the right thing to do and help them in any way collaborate. But I also think there's a lot we could do by participating, learning, blah, 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 blah. So I think we want to look for those opportunities.
Yeah, and I didn't really harp on the scientific outcomes, but certainly there are interesting new targets for drug therapies that are coming out of this. There are, new genes that we didn't know were involved in certain pathways because we didn't have the variants to discover that. There are new variants in known genes, but that are informative for function. So, there's a lot that will come out of all of this scientifically. I think Lisa has her hand up.
Yes, I just wanted to comment that thank you for your presentation, and it kind of gives new meaning to our terms of success and transformational, and that's really exciting. And building on that sort of new meaning to those terms, I wonder if there's any exploration about taking lessons learned on another continent where capacity was built and bringing that back home to institutions where we keep trying to help our colleagues build capacity for being prepared to submit applications to NHGRI or to other institutes. And I realize the contexts are very different, but I also wonder whether some of the internal will and transformation of what's going to of criteria and of what's going to count as success and what's going to count as capacity, if there are lessons there that could be valuable for sort of all of many of the topics we're going to be discussing today and tomorrow.
Yeah, Lisa, I 100% agree, and I really think that there is a lot to be learned about how you approach a less experienced institution, a lower resource institution, and how you think about what metrics they should be evaluated under, for sure.
Howard, did you have a question?
No, my question was answered. Thank you.
Okay, great. Tim.
So yeah, this is wonderful, and it's impressive to see how much has happened. Have you thought about maybe expanding beyond Africa? I mean, the lessons
Absolutely.
learned in Africa could easily go to lots of countries across the world.
Yeah, absolutely. I mean, I think really it is at least the focus of some of our thinking is shifting towards, again, the idea of low resource settings, of how can we increase reach? How can we ensure equitable access so that more discoveries are made?
Okay, thank you, Jen.
Thank you.
Congratulations. A lot of hard work.
And thanks to everybody who worked with me on this, because it was absolutely an amazing village.